Hello students, today we'll start with the topic embryogeny. What is embryogeny? Embryogeny is the formation of embryo from the zygote. So embryogeny involves formation of embryo from zygote. See students, this we have discussed earlier also that endosperm formation precedes the embryo formation. Why is it so? Because formation of embryo needs nutrition and that nutrition is provided by the endosperm. So in most of angiospermic families, endosperm formation is initiated before division of zygote to form the embryo. This will help in providing assured nutrition to the embryo. This line is written in NCERT and this question also comes in the board exams. Now we will discuss embryogeny in dicots. The embryogeny in dicots, it is given in NCERT. It was first observed in or studied in Shepherd's purse or Capsella bursa, which belongs to Cruciferae family, and therefore it is known as crucifer type of embryo development. See, here I have shown the structure, the changes which occurs in the embryo sac after double fertilization. This is larger cell is primary endosperm cell and this is zygote. This is micropylar end and this is chalazion end. So what happens? One transverse division will occur, one transverse mitotic division which is unequal occurs in the zygote. Two cells will be formed, one towards micropylar and one towards chalazion end. The cell formed towards the micropylar end is known as larger cell. It is larger in size. It is also known as suspensor cell, basal cell or hypobasal cell. The cell formed towards the chalazion end is known as epibasal cell or terminal cell. It is smaller in size and it is also known as embryonal cell because this is the cell which will form the embryo. Now what happens? This larger cell or suspensor cell will undergo transverse divisions. This larger cell or suspensor cell will undergo transverse division to form a column of cells. This column of cell is known as suspensor. This column of cell is known as suspensor, and in dicots this suspensor contains generally 6 to 10 cells. The last cell of the suspensor towards the micropylar end is big in size. This cell is known as hostorial cell. This cell is known as hostorial cell or vascular cell. Its function is to absorb nutrition from the surrounding tissues. Its function is to absorb nutrition from the surrounding tissue. The cell of the suspensor which is near to the embryo is known as hypophysis cell. Is known as hypophyseal cell. Now this small cell embryonal cell will undergo two mitotic divisions which are perpendicular to each other to form four-celled stage. To form four-celled stage which is known as embryo quadrate. Which is known as embryonal quadrate. Now this four-cell stage will again undergo pericrinal division to form 8-cell stage. To form 8-cell stage. So this is 8-cell stage. It is also known as embryonal octate. This is also known as embryonal octate. Therefore, two tiers of cells are formed, one upper tier and one lower tier. Upper tier also has 4 cells, lower tier also has 4 cells. This upper layer is known as epibasal layer and lower layer is known as hypobasal layer. Just for your information, this is the diagram of a dicot embryo. It has two cotyledons and this is embryonal axis. Two cotyledons and this is embryonal axis. This is radical and this is plumule. Part of the embryonal axis between the plumule and the cotyledon is known as epicotyle. Part of the embryonal axis between cotyledon and radical is known as hypocotyle. Now, growth of the suspensor cell is faster than the embryonal cell. This suspensor cell will grow faster as compared to embryonal cell. What is the function of this suspensor? This suspensor pushes the embryo inside the endosperm so that this embryo derives nutrition from the endosperm. So function of the suspensor is to push the embryo inside the endosperm to take nutrition from the endosperm. Therefore, growth of the suspensor is faster. Now, we have seen that in octet embryo, two tiers were formed, epibasal and hypobasal. Epibasal tier will form plumule, epicotyle and cotyledon. Epibasal tier will form plumule, cotyledon and epicotyle. Hypobasal tier will form radicle and hypocotyle. Epibasal tier will form radicle and hypocotyle. Children, I have told you that the cell which is near to the embryo, the cell of the suspensor which is near to the embryo was known as hypophysis. This hypophysis will form the radical tip. This hypophysis will form the radical tip. So now this octant embryo which I showed you earlier will undergo pericranial divisions to form multicellular mass of the embryo. This multicellular embryo or globular embryo, this is also known as globular embryo. In this globular embryo, initiation of cotyledon will start. When cotyledon formation will initiate, globular embryo will change into heart-shaped embryo or chordate embryo. See, this is heart-shaped embryo or chordate embryo. Now, this heart-shaped embryo or chordate embryo will change into mature embryo. Now, you can see suspensor is small and embryo is big in size. This embryo, dicot embryo, has two cotyledons. It has plumule and radicle. So, what is the sequence of events? Zygote was formed. Zygote formed two cells. One was bigger cell and one was smaller cell. Smaller cell was known as embryonal cell. This embryonal cell will form tetrad stage, then octant stage, then globular embryo, then heart-shaped embryo, and then mature embryo. So this is the sequence of events in the formation of mature embryo, and this whole thing is given in your NCERT. Dicot embryogeny is known as meroblastic. This is very important from competition point of view. See children, dicot embryogeny is known as meroblastic. Why it is known as meroblastic? Because whole of the zygote is not taking part in the formation of embryo. Because whole of the zygote is not taking part in the formation of the embryo, bigger cell is forming the suspensor, whereas smaller cell only forms the embryo. See, suspensor and embryonal cell. That is, whole of the zygote is not used in embryo formation. Only part of the zygote is involved in embryo formation. This dicot embryogeny is also known as endoscopic development. This is also known as endoscopic development because this suspensor pushes the embryo inside the endosperm. Inside the endosperm, or this whole formation of suspensor embryo is taking place in the embryo sac. That is in the interior of the environment, favorable environment. Therefore, it is known as endoscopic development. Suspensor pushes the embryo in endosperm. That is, embryo is present in the interior in favorable environment. Therefore, it is known as endoscopic development. Now, embryogeny in monocots. This is not discussed in your NCERT. I am also discussing it in very short because it is important for NEET. Embryogeny in monocots. Sagittarian type of development. That was known as crucifer type of development in dicot. Now it is known as Sagittarian type of development in monocots, and it was studied in Luzula. See, what is the similarity between embryogeny of monocot and dicot? Both the development is meroblastic and endoscopic. Both in monocots and dicots, embryo development is meroblastic and endoscopic. Initial stages of development in both dicot and monocot embryogeny are same. That is, globular embryo is formed, then heart-shaped embryo is formed. Dicot embryogeny, this is a comparison between dicot embryogeny and monocot embryogeny. In dicot embryo, suspensor is 6 to 10 cells long, whereas in monocots, suspensor is generally consist of one cell. One cell of suspensor was called hypophysis, which was near to the embryo, and it was forming radical tip. In this, no hypophysis is present in case of monocots. 2 cotyledons in the embryo, only one well-developed cotyledon, that is scutellum, is present. In some cases, remains of second cotyledon called epiblast is present, and it can be seen in some grasses. The embryo of grass is drawn in your NCERT, and you can see a very well epiblast there. Plumule has a covering of hollow foliar structure called colupetile. In monocots, the covering over, leaf-like covering over, foliar covering over plumule is called colupetile, and similar type of covering over radicle is known as coleorhiza. Both this colupetile and coleorhiza are absent in dicot embryos. Both these colupetile and coleorhiza are absent in dicot embryos. See, here I have shown the diagram of monocot embryo. This is the longitudinal section of grass embryo, that is monocot embryo. See, this is the well developed cotyledon, and which is known as scutellum. This is single cotyledon called scutellum, and it is well developed. This is plumule. This part is plumule, and it is covered by leaf-like structure, foliar structure called colupetile. This is epiblast. Epiblast, the undeveloped second cotyledon. This is radicle, this is root cap, and this is coleorhiza, foliar structure which protects the root radicle. It is known as coleorhiza. Now, one question. If a hexaploid female plant is cross-pollinated with octaploid male plant, then what is the ploidy of primary endosperm nucleus and zygote? Hexaploid female plant is there and octaploid male plant is there. So, in female, the egg will be triploid because the female plant is hexaploid, and pollen grain will be, or male gamete will be 4n because male plant is 8n. So the ploidy of the zygote will be 4n plus 3n is equal to 7n. But ploidy of pen will be 2 female nuclei will be there, 3n, and 1 male gamete 4n is equal to 10n. So this type of question can be asked in NEET.